In this module, we will discuss the role of RNA interference and its analysis. You see that this is also very important to find out that how the different genes they are knocked out, how the different genes can be silenced, or how or vice versa. Now, knocking out the genes, that can be done by mutagenesis. One way, it is a laborious process and has so far been accomplished on a genome-wide scale only in the yeast, that a certain genes, they can be knocked out, they can be silenced, they can be removed by the mutagenesis, by making the certain mutants, and that has been done only in the yeast. Now, knocking down genes, there is another approach, and that is the knocking down genes by RNA interference, RNAI. So some more complex organisms, they are amenable to a simpler alternative of knocking down genes by RNA interference. By RNA interference, that's the RNA which interfere, and it knocks down. What does? It stops that gene which is in the complex organism, and that is through RNAi interference. For example, the nematode worm C. elegans is particularly susceptible to RNAi, which even affects the progeny of treated worms. That is, where with RNAi, the progeny, offsprings of the treated worm also are affected by this gene silencing. Now, it can reproduce parthenogenetically, which means that only one parent is required. Parthenogenetically production means that the same parent is a male as well as a female, and it contains fewer than 1,000 cells, and its whole genome has been sequenced. This is the advantage, because 1,000 cells and whole genome has been sequenced in the case of C. elegans. So early embryogenesis, that is the Bertie Schoenson and colleagues, observed the effect on early embryogenesis, the first two cell division after fertilization. So when there was early embryogenesis, means that the first two cell divisions were after fertilization. These scientists, they observed the effect of embryogenesis. And inactivation of 1668 genes by RNAi produced detectable phenotype defects. Of these 1668 inactivation of 661 genes, they gave reproducible defects in the first two cell division, and the rest gave defects at the later stage of the development by RNAi silencing method. But there is a problem with RNAi technique. If sometimes it fails to inactivate genes, that is the false negatives, so negative results are difficult to interpret. It will be very difficult to interpret the negative results when the RNAi, with the genes which it inactivates, they give the negative results in the case of RNAi. Now, mutagenesis strategy, on the other hand, produces also false negatives. and the mutations are detected only if they give clear phenotypes. So the mutagenesis strategy also produces false negative, as in the case of RNAI analysis, they also produce the false result or negative. Later, the researchers, they grouped the 661 genes, which were discovered earlier, according to their specific phenotypes. According to their specific phenotype, जिस तरह उनकी phenotype C, they grouped them together, and they found that the inactivation of about half, that is the 326 of the genes, they produce defect in embryogenesis per se. It means during the embryogenesis, that was the defect produced. While the remainder 335, they simply affected the general cell metabolism required to keep the embryo alive long enough to divide twice. So it means that when they group together the different cells which had the similar phenotype, half of them, they produce the gene defects in embryogenesis, while the remainder 335, they simply affected the general cell metabolism up to the second division of the cell. And when they carefully annotated the specific defects, they found that the former 326 genes into the defects in 23 aspects of embryogenesis, such as spindle assembly, that is the 9 genes which were there, sister chromatid separation, 64 genes, and it means that the specific defects that could be annotated and grouped in the forms of 326 genes, where 23 aspects of embryogenesis that was found out, and during the RNAI analysis. Now, in this module, we have discussed RNAi analysis and the mutagenesis defects which are there, and compare these two methods for silencing the gene.